Alzheimer's is a tough disease because it happens later in life. It may happen progressively over time, and the brain is a very inaccessible organ to study.
The point of this study was to develop a resource for researchers to be able to better study Alzheimer's disease and related dementias. The way we envisioned this was to be able to grow cells from the brain of a person who's died so that we can compare those cells and those cell lines and the stem cells that we can generate from those cell lines with the actual pathology that's occurring in that person's brain at the time they died.
What we've done that is very new is that we've used a specific tissue source from the brain that we can take at autopsy and grow and convert that into neurons.
The reason we chose the meninges to try to develop these cell lines is because they're bathed in the same fluid, the cerebral spinal fluid, that the brain is bathed in. And so we feel like the meninges are the closest tissue that we could get to be able to generate these cell lines that were representative of what the person's brain has experienced during their lifetime.
The only definitive diagnosis of Alzheimer's disease comes after autopsy, so we've been working with the neuropathology core at UW to get their definitive Alzheimer's diagnosis and then take cells from a small layer of tissue surrounding the brain that will grow after autopsy and use those to make stem cells from. So then we have an infinitely renewable source of patient cells from people who have a neuropathological diagnosis of Alzheimer's disease.
We do have great interest in understanding how the biological age of a person is different from the chronological age. And so people can be the same age on the calendar, but their bodies may age differently. And we're really interested in on how that impacts disease and Alzheimer's disease.
These patients are coming through incredibly well characterized longitudinal studies in Seattle. They've been followed for many years. Their clinical history is very detailed, their genetic history is very detailed, and now we'll have their cellular profiles as well.
The technology that Jessica and her collaborators have helped develop, which is to direct these cells directly into nerve cells, enables us to maintain those age-related changes and then perhaps be able to study the effect of aging and biological aging on disease.
In our lab, what we're trying to do is really understand how human genetic variation predisposes to Alzheimer's disease risk.
Why these cells are so important is because we think they're very representative of what's happening in the brain. And unlike a human brain, which we can't do experiments on, these cells enable us to actually do experiments. So we can perform science, which is hypothesis generation and then testing using experimentation in these cells to then ask, how does this disease happen? And then to ask, how do we diagnose it better? And what kind of drugs or other interventions can we bring to bear to stop its progress or even stop it from ever happening in the first place?
We're not going to regenerate the whole brain, and we're not, making cells that could be right now transplanted back into somebody else's brain. But we are basically taking neurons, making neurons from deceased people to understand biological pathways that might have gone wrong that led to disease, using these cells to test compounds that may be beneficial for people, even maybe their family members who may develop the disease.
Humans are so widely varied. There's so many different things that we have exposure to during life. The different things that we do, our different genetics, and to really understand the kinds of effects that may be driving disease, we need to evaluate large numbers of subjects. And our hope is that over the next few years, we will generate ultimately hundreds of these cell lines from these subjects to be able to really inform the science.